Okay, hello everyone. I would like to warmly welcome you to this open discussion forum of the 75th anniversary of Albert Szent-Györgyi's Nobel Prize Award. And let me extend a very warm welcome to our Nobel laureates sitting here in the first row and thank them for accepting our invitation. Thank you very much. My name is Silvia Krizo, and I have been asked to host and moderate this event, which is a great honor and a big challenge for me too. To tell the truth, I have absolutely no idea about the latest scientific research. So when you start asking questions about, I don't know, peptide hormone production of our brain or other of specialist area of our Nobel laureates, I will definitely lose the thread. Please don't do that. But you can ask any questions you would like to ask. Let me say a few brief words about today's program. The plan was that firstly 9 secondary school students will introduce our Nobel laureates one by one in the order in which they have received the Nobel Prizes. The students come from 3 Hungarian countries and also from Serbia, and they won a multi-round contest organized by the University of Szeged. After this, there will be a session for questions. 27 people were previously chosen to ask their questions, and they have attended at this contest too. But if you have any questions, please really don't hesitate to ask these questions. And as I mentioned, that was only the plan, because sometimes, not sometimes, the things never go quite to plan, so we have made some changes. We will start the introduction of Yonath, who has received her Nobel Prize in 2009. Because of her schedule, she has to leave us soon. So we will do her introduction, and after that introduction, you can ask your question, and then we will continue the introduction of our other Nobel laureates. And then I would like to ask, sorry, I have to look for who will introduce Yonath. So please welcome Yonath, who has received her Nobel Prize in 2009. She is there in the 4th row. And the introduction to her is by Paula Dietrich, representing Tömörkény Secondary School, Szeged. Please welcome her on the stage. Maybe may I ask you to join us on the stage? Thank you very much.
Ladies and gentlemen, it is a great honor for me to be able to introduce to you Ada Yonath. She was born on the 22nd of June in 1939 in Jerusalem, Israel. She graduated from the Hebrew University of Jerusalem in 1962 with a bachelor's degree in chemistry, and in 1964 with a master's degree in biochemistry. In 1968, she earned a PhD in X-ray crystallography at the Weizmann Institute of Science. She accepted postdoctoral positions at the Carnegie Mellon University and MIT in the USA. She received the Nobel Prize in 2009 along with Venkatraman Ramakrishnan and Thomas A. Stites for her pioneering studies on the structure and function of the ribosome. She became the first Israeli woman to win the Nobel Prize out of 10 Israeli Nobel laureates, the first woman from the Middle East to win a Nobel Prize in the sciences, and the first woman in 45 years to win a Nobel Prize for chemistry. She focuses on the mechanisms underlying protein biosynthesis by ribosomal crystallography, a research line she pioneered over 20 years ago. For enabling ribosomal crystallography, Ada introduced a novel technique, cryobiocrystallography, which became routine in structural biology and allowed intricate projects otherwise considered formidable. She is currently the director of the Helen and Milton A. Kimmelman Center for Biomolecular Structure and Assembly of the Weizmann Institute of Science. And without further ado, let's welcome Ada Yonath.
Thanks a lot. Were you satisfied with this introduction, or would you like to add something? You have your seat here. Please take a seat if it is much more comfortable for you. Yeah? Okay. How you would like to... Okay. So, ladies and gentlemen, you can ask any questions you would like to have. Let's move to first question. Who would like to ask her? But please start the questions because Ada has to leave us. There are some questions in the second row, please. The microphone are for our students.
Good afternoon, everybody. My name is Anna Hoffman from Darfrant High School, and I would like to... So my question is that, how did your life change after you received the Nobel Prize?
It doesn't work, or maybe it does. Do you have a microphone here? Yeah, okay.
Well, one thing that changed, I now know to look for microphones and to make them work, which I was not so familiar with before this. I think that my personal life did not change. I'm still in the lab, I'm still working, I'm still interacting with students and with professors. My home, my family, my interactions with friends did not change. But the possibility to talk to young people that want to know about science, this did change a lot, and I'm very happy about this. This is the best thing that could have happened with the prize, that there are so many here and some other places that want to know what does it mean to be a scientist, or what brought me, whatever question, because I think that in this way I interact with the future, not only with the past, and maybe I can even change it to the good. So this is the main change. There are some other changes that I don't want to talk about because they are not so pleasant as today.
Okay, we can move on to the next question if somebody has got another question. Actually, were you a really diligent student at school? Were you a really diligent student at school? Yeah. I don't want to hurt you.
What do you mean by this? Did I get good marks? Could I repeat correctly the teachers? Or did I do my homework? What is the question?
Yes, for example, in this order.
I was considered a good student. I'm not sure that I repeated what the teachers said. And what was nice at my time, the teachers encouraged us to ask all types of questions that have to do with curiosity, and I enjoyed it very much.
Has got anybody a question? Very good. In the second row, please. Okay, here's my microphone too.
What was your main motivation to carry on with your work?
What was my main
motivation to carry on?
Motivation? Oh, I wanted to understand. Curiosity. That's in one word, this is the word. I was curious. I wanted to understand many things about nature, and I was very lucky that I could work on what I thought was important and interesting for me. But curiosity was the driving force, and passion.
Other questions? Yes, I see a hand there.
What are you doing in your free time? What is your hobby?
What's my hobby?
Yeah.
Well, there is a big difference between hobby and free time. Free time we don't have so much. But we are trying to get as much as possible. So what do I do when I don't do science? This is what you ask. So first of all, I want to tell you, as young people, you may like to know, being a scientist is sometimes working on your hobby. It's not sometimes, it should be working on your hobby. Even if not everything is going like a hobby, and even if there are some very weak minutes, and even if there are some routine work that is really boring, the whole outfit and the whole dream is your hobby. And I think we as scientists are very fortunate to be able to work on our hobby and to exist like that. But on top of it, sometimes I read. I like swimming. When I was younger, I was doing work with my fingers. I think that I can do it now too, but I have less time. And what my dream is to write. I have some ideas inside me, like novels. I never got to do this, but this is maybe when I retire, I'll do that. And I have friends.
We can move on to another question if you would like to ask. Actually, can you remember the first moment when you decided to become a scientist? Or is it not a normal question to...
No, the question is good. The answer is not so great. The question is normal. I didn't decide to become a scientist when I was more or less your age. This is because I didn't know that there is a profession called scientist. I just didn't know this. I was raised in a poor family with hardly any connection to science. But my parents let me go the way I wanted. My father died early, my mother did. Continue letting me do what I wanted. I actually wanted to go and do something for my country, which was at that time agriculture far away. My real dream was to raise cows. And actually, from the beginning. And I even had the exact way to do that. But because, as I told you, my father died when I was really young, before 11. There was a baby at home. My mother was not so well, so I had to help. And when I finished high school, I still had to help. And I couldn't go with my cows somewhere far. So I decided until I can go and do this, I will do something that is interesting, which means studying. So I knew that there are universities, and I knew that one can study. But I didn't know that there is a profession of doing science. I thought that the professors at the university are teaching, and the people that study there can become afterwards teachers themselves, or go to the industry, or go to hospitals, do things that they studied. But I didn't believe that somebody can work on a problem that is interesting for, and somebody will pay for it. This was above and beyond me. So when I was a student already, in the second year, I think, the teacher taught something, and I had a question about it. So I asked the teacher in the intermission, what does it mean, what you said? I don't remember the question, I don't remember the answer, but I remember that he said, oh, that's exactly what we are now investigating. Come to my laboratory, and we will discuss it with the student. And then for me, it was something really new. There is laboratory, he can do science, he has a group, and he can work on the problem that is interesting for him. So I thought maybe I will do this too. Is this a good answer for your question?
It was a very nice answer.
So
Thank you very much.
I'm not sure that this is the way everybody has to go, but this is what happened to me.
Thanks a lot. I don't know, I know your schedule, and I know that now you should leave, but if you would like to stay, we are very happy.
Thank you.
Okay. Do you have any other questions to Ada Yonath? Okay, then I would like to say thank you very much for answering our question and to attend at this occasion. Thank you very much.
If you want to hear an advice from me, don't necessarily go to science if you don't like it, but do what you like to do and you can do best. Do what you you really feel that you fit for this and it's really interesting for you. And if you go to science, I'll be even happier. Thank you.
Thanks a lot. Thank you very much. And then we continue with our individual introduction. We're pleased to welcome onto the stage Robert Huber, Nobel Prize in Chemistry 1988. Please take a seat. And the introduction to him is by Daniel Schinka from József Attila Secondary School, Mako. Please welcome him also on the stage.
Ladies and gentlemen, it gives me great pleasure to introduce the Nobel laureate Robert Huber. He was born on the 20th of February 1937 in Munich, Germany. At the age of 10, he began attending the Karls Humanistic Gymnasium in Munich, where the basic subjects were Latin and Greek, with little natural science and only a few hours of chemistry a month. However, he learned quite easily and spent his free time doing sports and satisfying his interest in chemistry by reading as many textbooks as he could. After finishing the gymnasium, he was accepted at the Technical College in Munich, where he began to study chemistry more formally. He graduated in 1960 and studied crystallography under W. Hopper, gaining his PhD in 1963. Even as a young graduate, he noticed some mistakes in an experiment by a Nobel laureate and was able to correct them. This, together with the discoveries he made in the field of crystallography, encouraged him to devote his life to science. In 1976, he became a director at the Max Planck Institute for Biochemistry in Martinsried, Germany, where he remained until his retirement in 2005. During his long and distinguished career, he has not only advanced the field of crystallography and structural biology, but also developed new methods of research and computer programs which are used in laboratories around the world today. In 1988, he was awarded the Nobel Prize in Chemistry together with Johann Deisenhofer and Hartmut Michel for the determination of the three-dimensional structure of a photosynthetic reaction center. Since retiring, he has taken up posts as guest professor at universities in Germany, Singapore, Spain, and Wales. He is an editor of the Journal of Molecular Biology and member of several prestigious academies. Ladies and gentlemen, please welcome Professor Robert Huber.
Only one question. Is there anything else you would like to add to this introduction, or you are very happy with that? Were you?
Well, I'm happy with it, and I would like to leave time for questions, time for the students to ask me.
Now, you would like to have your questions?
Yes,
okay.
Of course.
Then let's do that in this way. Okay, ladies and gentlemen, you have now the opportunity to ask your questions. Who will ask the first question to Robert Huber? Okay, in the second row, please.
My name is Katalin Mango, and I am from Tömörkény Secondary School. And my question is, have you ever thought about trying another field of science, such as, I think, physics or someone else?
I had no chemistry at all in school. I was perfect in Latin, fluent in Latin after my 9 years of gymnasium. I had Greek, which was a nuisance. Greek I forgot immediately after school. Latin I kept, and it helped me with the Romanic languages very much. This I would like to recommend still for teaching in schools. So I taught myself chemistry because I had no chemistry teaching. There was in the last few years of my gymnasium, there was a few hours of chemistry teaching by our biology teacher. So I went there, and I clearly knew much more than this guy did. And I interrupted him frequently, said, "No, this is not correct," and he then took me aside and asked me not longer to visit his teaching. So this was my, I was quite decided to do chemistry. Now, how did I come to crystallography? Well, that had to do with my inclination for sport and mountaineering and skiing, going to the mountains. Now, Munich is just a few kilometers away from the mountains, the Bavarian, Austrian mountains, and when you climb there or walk there, then, and do not only look in the distance at the next mountains, but look down, then you see minerals, you see crystals, some of them very beautiful, colored, shining faces. So I collected, and then thought I should study them in more depth, and the only way to do that is by crystallography. So I asked Walter Hoppe, who was one of the very few crystallographers. Now we are talking about the 50s, so crystallography was not what it is now. Tools had to be developed, and there were not many people in Germany and not in the world who really were experts in crystallography and were able to develop methods. So I entered his group and learned crystallography. And then, well, I did a small organic molecule structure, but then I met a biochemist, Adolf Butenandt, who was at the university in Munich. Now, Butenandt was a famous biochemist for his discovery, this work on, and his discovery of the chemistry, the molecular structure of the human sexual hormones. He received the Nobel Prize in 1939 for his work. Well, just an anecdote illustrating the times in which Butenandt lived. He was not allowed by the Nazis to go to Stockholm to receive his prize. So he just was not given a visum. He was not allowed to do that. So he received medal and documents then after the war in 45, 46. Well, there is another story to tell about the situation with Nobel Prizes and Nazis, which had to do with another laureate whose centennial we now celebrate 2012, and this is Max von Laue. He discovered X-ray diffraction with this seminal experiment in 1912, in that way proving that X-rays, Röntgenstrahlen, are electromagnetic radiation, and crystals are three-dimensional ordered lattices. So this single experiment in 1912 was a proof for these two key questions that we had in material science and in physics. And of course, it was the start for the use of X-ray crystallography to determine molecular structures. Finally, protein structures, of course. This is the theme of my lecture tomorrow, the use of crystallography for determining large protein structures. But now back to how did I come into biochemical problems at that time. So I met Adolf Butenandt, and he had just discovered that insects do have hormones. Now, up to that time, insects were thought to be hormone-less animals. But we know that insects undergo many developmental stages, so different pupal stages, then the final stage, the imago. All of this happens, but people thought it happens in some mysterious way. Now, Butenandt and his associate, Peter Carlsson, found that it is hormone-driven, and they isolated the hormone, and the hormone is ecdysone. I think if you read textbooks about insect biology, you will stumble across ecdysone. So there was the ecdysone, but there was no real way to determine its three-dimensional structure. There was no mass spec at that time. The molecular weight was unknown. So they determined molecular weights at that time by, now I ask, by ebullioscopy. Students, does anybody know what this is? Is there a hand up? No. Well, it's a tricky experiment, but you measure the seeding point elevation or the freezing point depression when you dissolve a substance in its solute. It's a tricky experiment, and you make errors. And Butenandt made an error. So the molecular weight was wrong, and it was wrong by a factor of 2. Now they had carbon, hydrogen, oxygen analysis, all of this. And from this they concluded ecdysone should be a naphthalene compound. So very different from the human hormones. Now I came in, crystallized it, and by just determining the cell size of the crystal, the density, an easy experiment, you can determine the molecular weight. And I found a value which was double that of what a Nobel laureate had found. Now, what to do? I was a young guy of 22 or 23 years. Now, how to approach a Nobel laureate and tell him that he's wrong? I didn't dare that. I did not believe in what I had, although it was a very simple experiment. Now, finally, I remeasured the unit cells and the density. And after 14 days, and I don't know how many times I remeasured it, I thought, I am right, and this guy is wrong. So this is how I become a scientist, I would say.
Thank you.
Right, so that was a long answer.
No, but it was a very nice answer, and in which there was a presentation too. Thank you very much for that. But maybe we have got another questions too. I hope that we have got another question. There is, yeah, in the first row. Do we have a microphone? Yeah. It's a very nice cooperation.
My name is Adam Sirer from the Tömörkény István Secondary School, and I would like to ask, did the way change people speak with you or people interact with you after you received your Nobel Prize?
Well, I think Ada already said her view, which on this point. So one of the nice things with the Nobel Prize is the much more frequent interaction with students, which I like very much, both young university students, but also school Well, this, I think, was for me, as I see it now, the main difference to the time before the Nobel Prize. Otherwise, there was relatively little change. I had a large research group before the prize. I think I was known before the prize, perhaps a little more known to politicians after the prize. Well, they did not listen before and after to me. Anyway, well, was this an answer?
Yes, yeah, thank you.
Right.
Okay, we can move on to another question. Who would like to have this next question? Actually, I've got one. There are many scientists who work in parallel on the same topics. What does it depend on that one of them will win a Nobel Prize and the other won't?
I did not understand.
There are many scientists who work in parallel on the same topics maybe, but for example, you have received the Nobel Prize, but many other scientists haven't received it.
Yes, that's
what is depend
true.
on? Yeah.
So there are many more who also would deserve a Nobel Prize and do not receive it. So there is a certain substantial degree of luck behind there too. Now, in our case, the work that we did on the photosynthetic reaction center was published in '85, and the Nobel Prize was awarded in 1988. The work was unique in a number of aspects, and there was not really competition at that time. Competition, so parallel work or work on related structures then came later, building on what we had determined. Though this was not a real problem in our case, it was, again, it was work that was unique for several reasons. One was the crystalline membrane protein, the first of its kind, and the other one was that it explained photosynthesis, so the biological process on which we all live, humans and animals all live on plant food and, of course, on the oxygen that we breathe. Well, so competition did not play a role, but I'm of course aware of many colleagues who received or did not receive a prize where competition played a role, and this aspect of luck comes in. Right.
Thanks a lot. Can I have another question, or would you like to leave now? Would you like to have it? It's here. Okay. My question is, do you often ride bicycle?
I always ride bicycle. Of course, at home, when I'm in Munich, I go from my house to work, which is 15 kilometers one way and 15 kilometers the other way. So I usually bike for 30 kilometers when I'm in Munich, and wherever I am, and I'm traveling a lot, I try to get a bicycle. Also in Szeged. I have my bicycle.
Yes, I know
here.
that. I realize that. Okay, but I realize that our student has got another question. Please don't hesitate to ask it.
In a previous Nobel laureate meeting, you said you are looking for the new Einstein of biology. And do you think you will find this new genius, maybe if I can say that, on this meeting, or have you found him or her yet?
I don't think I did understand your
On a previous Nobel laureate meeting, you, in an interview, you said you are looking for a new Einstein of biology.
Oh, yes. This is what I said. Well, I haven't found one, but maybe there is one. So the question I was answering was, there is a basic biophysical problem that we have, namely the folding problem. So we do have in protein structures, which emerges from the ribosome as a flexible polymer chain. And then it folds up spontaneously or with some help by chaperones or chaperonins to a well-defined, usually, in most cases, well-defined three-dimensional structure where each atom, each of these 10,000 or even more, 50,000 atoms knows its specific place in this very irregular structure. This is a mystery. It's a fantastic problem. Because we know it happens. So there is a rule. Why do we not understand it? Why can we not simulate protein folding in the computer? Why do we have to do the experiment? Why do we need crystallography? We have to do the or NMR or electron microscopy because the code for the structure is written in the amino acid sequence. And nature knows how to do it. And it happens millions of times while I'm standing here in front of you. So this is the problem. Well, there is slight progress to understand better, but I do not really see light in this dark tunnel. And maybe it's just an insoluble problem. It's just too difficult, too complex. Maybe it's a basic new idea which is missing. And this is what I meant with the Einstein, that a person who perhaps sees this perhaps very simple basic idea and solve the folding problem, then we do no longer need any crystallography, any electron microscopy. We do not need NMR. What do we need at all? I don't know. So this is what I meant.
Thank you. Thank you very much.
Okay. Do we have another question? Yes, in almost the last row, I cannot count in which. Can we have a microphone there, over there, please?
I may try to speak up.
Very loud, please, because we don't hear it.
My name is from the pharmacy faculty. It was just very nice to hear that your research career started with ecdysone research, because so did mine. So I may have some bright future. That's not what I want to ask. I would just like to ask you if you had any further progress on that field of research. I mean, with this insect molting hormone stuff.
No, I did no longer work on it. We worked on plant proteins, but not on the ecdysone receptor, for instance, which is known. Meanwhile, the extracellular part, at least. So no, we did not work. It's still of the receptor. No. Yes. Didn't I tell that?
No, you didn't say that.
Oh, I'm sorry. Well, I'm so proud of that. No, it was in all beautiful details. It's a steroid, and it has noble substituents. So it is related to the human hormones. So it showed, I think, very early, for the first time, this molecular evolution in the hormone family. What year was that? I published that 1963. It was my thesis or postdoc work.
Okay, thanks a lot. I think it was a historical moment that two Nobel laureates discuss the same topic here in this room. It's very nice to hear it. Do we have another question, or do I have to say thank you? Okay, then I would like to say thank you very much for your answers, and let's continuing with the introduction. Okay, it's a pleasure to welcome Bert Sakmann, Nobel Prize in Psychology or Medicine 1991. Please welcome him onto the stage. Or? I'm sorry to have to say, but he's not here. But we will introduce him, okay? Are you agree with that, yeah? I would like to ask Dorina Nagyszűcs, representing the Bibor Secondary School, Kiskunhalas, to introduce to us Bert Sakmann.
Ladies and gentlemen, it is such a pleasure to be here and have the opportunity to introduce you Dr. Bert Sakmann. Bert Sakmann is a German scientist who was born on the 12th of June in 1942 in Stuttgart. As a child, he was interested in physics and dreamt about becoming an engineer. However, at his first university in Tübingen, he learned medicine. Later, he decided to study further in Freiburg, Berlin, Paris, London, and Munich. In these places, he met several people, for example, Sir Bernard Katz and Erwin Neyer, who made a huge impression on him with their research projects. By the time he became a member of staff at the Max Planck Institute for Biophysical Chemistry, Dr. Sakmann was already a specialist in the field of ion channel physiology. With Erwin Neyer, they were able to prove the existence of the ion channels and discover their significance for the human body using their breakthrough, the patch clamp technique. This method can be used for measuring the currents passing through the ion channels of the cell and the activity of voltage-dependent channels. For their work in this challenging field, they were awarded the Nobel Prize in 1991. Their discovery of how to study ion channels promoted the development of different medicines, such as heart medicines that act on ion channels. At present, he is working at the Max Planck Institute in Florida, and he focuses on the functional anatomy of the circuits in the brain that are responsible for the simple behavior patterns. He is also trying to make a three-dimensional map of the normal rodent brain. With such research, his aim is to carry out studies on brain degenerative diseases, such as Alzheimer's. Ladies and gentlemen, please welcome him if he arrives.
Thank you very much. Thanks a lot. And we would like to introduce Eric Wieschaus, Nobel Prize in Psychology or Medicine 1995. The introduction to him is by Eszter Nógrádi Deák Secondary School, Szeged. Please welcome her onto the stage.
Ladies and gentlemen, of all the species of literary composition, perhaps biography is the most delightful. Robert E. Hall quoted, "Like many of you, I realize that the chance to meet our guest in person today is a rare and extremely valuable opportunity. It is both an honor and a privilege to introduce a man of distinction to you, Eric Wieschaus. Eric was born in South Bend, Indiana, just after the Second World War. As a child, he spent countless hours drawing and painting, dreaming of becoming an artist. Today, he studies shape, movement, and form, not on canvas, but in developing embryos. Eric was introduced to Drosophila as an undergraduate at University of Notre Dame, where he could first see cleavage and gastrulation of living frog embryos. He immediately wanted to discover the genetic background of developmental events. Eric decided to begin graduate school and earned PhD in biology at Yale University, where Donald Paulson, a Drosophila geneticist, had a place set up in his laboratory for him. In 1978, Eric began collaboration with the molecular biologist Christiane Nüsslein-Volhard in Heidelberg, Germany. Since 1981, when he moved from Heidelberg to Princeton, he has been teaching genetics and development. In 1995, Eric won the Nobel Prize in Physiology or Medicine, along with Edward Lewis and Christiane Nüsslein-Volhard, for their work on identifying key genes that make a fertilized fruit fly egg develop into a segmented embryo. Today, we have the pleasure to have him here to tell us more about his scientific discoveries. Please join me in welcoming Professor Wieschaus.
Okay, thank you very much for this excellent presentation. And I have to ask you, what do you prefer? To answering your questions now or to listening to other introductions? Oh, it's the first question.
It's a hard question.
Because I feel alone here on the stage. I would like to be very happy if many Nobel laureates would sit on this seat. But if you would like to answer the questions now, of course you can do that.
The difficulty for a scientist is standing on a stage and having people look at you. And it's much more comfortable sitting out in the audience.
Oh my God, it's terrible to hear that, but okay.
I'm shy, so we'll do my questions and then I can go back.
Okay. Okay, let's do that, please. Let's hear the first question. Yes, I see the hand there over there. And please take the microphone as quickly as it's possible. Thank you.
I am Lila Sivic from Tancic Mihai Grammar School. Can we apply the learnings of your studies on the Drosophila to human development?
Okay, so the question is, in part, how similar fruit flies and fruit fly embryos are to human embryos. And for me, that's an especially important and good question, because I have to tell you that when we did the experiments that led to the Nobel Prize, we thought we were doing experiments on fruit flies. We didn't know how similar fruit flies would be to humans. We did the experiments out of curiosity and because we wanted to understand how any embryo developed. We thought that would be helpful for medicine to know how any embryo developed, but we never anticipated the direct application of our experiments to human biology. In retrospect, that seems kind of stupid. If we had thought about evolution, we would have realized that once genes evolved, and once they were made proteins that were functional, those genes would be maintained throughout all of the life forms on Earth. We didn't realize that when we did the experiments. It was later scientists who developed molecular biology techniques to clone genes and to recognize the similarities between humans and fly genes. So there are really two lessons there. I think the first one is the wonderful feature of biology that we share with all life on this earth, common strategies and common genes, and that means that as scientists, we can follow our curiosity and be relatively certain that it will have an impact on our life as humans, on medical issues. There's a second, more personal lesson, and that's that as a scientist, you almost never know the importance of the experiments you're doing at the time that you do them. You do them because of curiosity. The importance that those experiments achieve are from, those experiments derive their importance because other scientists pay attention to them and build on them. And they build on them in ways that you never anticipate, that you never expect. And that's truly one of the great things about being a scientist, that other people, other scientists, build on your experiments and your knowledge.
Thank you very much. We can move on to another question. Yes, please, in the first row.
So I'm Fanny Price from Mora Ferenc Secondary School, and my question is that do you still spend your free time on drawing and painting, or do you have any other hobbies?
So do I spend my time drawing and painting? It's a curious question. There was a time in my life where I painted intensely and I drew and I really wanted to be an artist. And then I became a scientist, and because days only have 24 hours, I had less and less time to paint and to draw. In a certain sense, all of my science, though, has remained an artistic effort because it always involves looking at biological material, photographing, documenting. I draw all the pictures in the lab for scientific publications that we do. What is interesting, though, is just last year, I went to the store and bought a large supply of paints. And I've not said this in public at all. This is terrible, very revealing. But I've actually started to paint again. And it's a very curious experience because some things you lose with time, and other things, other skills you have, you still find somehow in your hands. And so for me, painting right now is a new experience in part, but it's an old experience. It kind of builds on things I could do. One of the interesting driving forces for me as a painter right now is something that I've never done before as a painter. I discovered a box of old photographs of my parents and my grandparents, and wonderful family photographs from 50 or 100 years ago. And these were on paper and they were decaying. And one of the triggers that made me want to start to paint is I wanted to take those photographs and somehow transform them into paintings that would be different, but in some way would be the same. Because in a curious way, I wanted to give those fragile photographs a longer life. I wanted to give them something special. So it's always with artists that are people who want to be artists, that there's some hidden emotional desire, something that drives, something very special about the particular project. And so right now, I started painting again, and I started painting in part because I wanted to be an artist again, but also for more personal reasons.
Thank you very much. I'm sure there are plenty more questions, for example, there. Thank you.
My name is Adam Mészáros, and I'm studying at the University of Szeged in biology. My question is, we are well aware of how conservative master control genes are, and they suggest homology. If we change two of these genes between, for example, a fruit fly and a frog, could we get an animal which can develop and live as usual?
Okay, so during evolution, genes and proteins stay essentially the same in their functions, but often they evolve subtle differences. So one of the very hard experiments to do is to take a gene from one species and put it into another species and have it work as well as the gene itself, as the normal gene would have. So we've, in general, have had a mixed success whenever we switch genes from one species to the next, in that proteins kind of work, and they help us to understand how similar things are, but they never work exactly the same.
Would it modify the organogenesis?
Would I say that? Ask the question one more time.
By knocking the genes.
Knocking out. Or
knock in.
Yeah, the genes.
So
right. Yes. So
would it modify the organogenesis?
Could you? Yes. We would. We've done that. It works sometimes. Mm-hmm. Sometimes it doesn't work. And the experiments, the results have been complicated enough that we have actually stopped trying those kinds of experiments. Just simply because it's too hard to interpret. The success or failure is always somewhere in the middle. Okay?
Thank you.
I hope that's a clear answer.
Who would like to ask? Oh, Tim Han. One Nobel laureate ask another one.
Another question.
Nobel laureates ask hard questions. We'll see.
No, I was just sort of curious. So some of the genes that I guess you and Yanni discovered turn out to be rather important for human cancer. Were you surprised when that happened?
We were looking for genes that controlled embryonic development, that were needed for normal development, told the cells what they should do, when they should do, how to do things in an embryo. What is surprising, we didn't anticipate, but again, if we'd been smart, if we had been the Einstein that you're looking for, we would have anticipated that the genes which are required in normal development are genes which are very powerful in their effects on cells and whose activities have to be highly regulated. And we know now that once you become an adult, many of these genes are not used or are used in very limited ways. Cancers, many cancers arise because these genes which are required in the early embryo, in a regulated way, become active inappropriately, in the wrong way, in our adult bodies. So it's the same genes, the genes that we need in an embryo and the genetic pathways we need in an embryo are the same pathways and genes that later cause us problems with cancer. It's inevitably going to be that way. The genes have power. They have biological functions which are essential. And we have become as complex as we are as humans because we have these wonderful complex pathways. In a way, it's not surprising then that as humans, we have such a problem with cancer. Because the same things that we benefited from, the genes that we benefit from as an embryo, become problematic as activities later in development.
Was this answer quite okay for you? Okay.
Was it good enough?
That was fine. Yeah. I mean, the interesting thing, I guess, is that in the embryo they make these beautiful patterns. I mean, so stereotypical and so beautiful. And in the cancer is precisely the opposite. I mean, they're just so ugly and disorganized.
That's because there is natural selection and millions of years of evolutionary selection on the pathways and on their control in an embryo to guarantee that that embryo develops into a beautiful structure. There is no selection on making a beautiful tumor. And so you see the abnormal morphology. Actually, we reverse it. You can say the extraordinarily beautiful morphology that natural selection and evolution has given us through these pathways and through these genes.
Thanks, Yonath. Does everyone have another question? Or yes, the last question to Eric Wieschaus over there, please.
Hello, my name is Miriam. I'm from the Szeged Medical School, the German program. And my question is, we have this huge upcoming stem cell discussion. And what do you think? Is this the future of medicine, or is it a risk?
Ask me what's the thing that's coming?
Stem cells.
Stem cells. What is the value? I think I am very optimistic about stem cells. The possibility of making stem cells, I believe that it will be an extraordinarily useful tool in medical biology. You notice I said medical biology, I didn't say clinical treatment of patients. I believe with great certainty that the ability to make stem cells and to test gene activities and to test cellular activities in stem cells is a powerful tool that will help us understand human disease, will help us to understand cancer, will help us to understand many biological abnormalities, much of our own biology, and will ultimately contribute greatly through that understanding to medical treatments. I think it's a bigger challenge for any of us to predict the practical uses of stem cells in any particular therapy. Yeah, could we grow a new pancreas, for example? We certainly can't now. I think it's in the realm of the scientific possible, and therefore I would support the research that would attempt to do that. Am I certain that that's the route that stem cells will be useful? No. But I think that the technique is so powerful that as scientists, it's always useful to try to see, can we use it here? Can we use it here? Can we use it in basic research? Can we use it in understanding nerve conductivity? Can we use it in any of these areas? And so that's where I would put stem cells right now.
Thanks a lot. Ladies and gentlemen, Eric Wieschaus, please welcome him. Thank you. You can take your seat if you would like to. And ladies and gentlemen, please join me in welcoming Peter C. Doherty, Nobel Prize in Psychology or Medicine, 1996. The introduction to him is by Borbána Szepes, representing Ságvári Secondary School, Szeged.
So let me start by saying how happy and honored I feel to have this chance to introduce to you all today Peter C. Doherty. He's a pioneer in immunology, and he represents the significance of medical research on the public stage. He has also written 2 books of his own, one of which is a semi-autobiographical one, giving advice on how to become a Nobel laureate. And as I've come to know an hour ago, he's planning to publish 2 more the next year or so. So his innovative personality, his commitment to amend humanity, not to mention his undeniably revolutionary efforts and achievements inspired me to study his life. I try my best to summarize it as thoroughly as possible. So the very fair-skinned man of Irish origin was born in Brisbane on the 15th of October in 1940, where his early years were motivated by the values of his 2 grandmothers and his father's encouragement to seek a life motivated by the willingness to learn. Then, incited by an attractive technician, he decided to study at the Veterinary University School in Australia. And later on, he researched in Edinburgh at the field of experimental pathology in Canberra, and later on in Philadelphia too. As you may know, he received the Nobel Prize jointly with Rolf M. Tinkernagel in 1996 for the discovery of the immune system's unique protection against viruses. To put it briefly, they inoculated mice with a virus causing meningitis and isolated the immune T killer cells, which were responsible for the recognition of 2 things on the membranes of the infected cell. The virus antigen and the MHC molecule, which before had been thought to be responsible for the rejection of cells during organ transplantation. As for his current projects, Peter supervises research into a new swath of flu viruses, not to mention his $300 million a year work, which aims at developing an HIV vaccine. Last but not least, this innovative personality has managed to trigger care and interest from his audience on his several media appearances, speaking up about worrying issues of our times, such as environmental degradation. So Peter's managed to capitalize from his fame and became a role model for young adults and to everybody. So please welcome Peter C. Doherty to this stage.
Would you like to say a few words before asking the questions, or?
Yeah, thank you. That's a very nice introduction. Thank you very much. Unfortunately, our grant support is more like 300 million forints than $300 million. So it's not quite that much money. I wouldn't know what to do with $300 million. I mean, I would just make a fool of myself. Yeah.
Okay, let's move on to questions. Let's hear the first question, please. There in the second row, I see. Thank you.
Good afternoon, everybody. My name is Diana Badinsky, and I'm from Wieschaus Secondary Grammar School. And I would like to ask that what was the most interesting finding during your research?
Oh, the finding that won the Nobel Prize was our biggest finding, and it was, it certainly was totally fascinating because we made a discovery. You know, Nobel Prizes come for different types of things. If you've heard from 2 X-ray crystallographers, Ada Yonath and Robert Huber, and they can decide to solve a very difficult problem. And they can go ahead, and if they're very clever and have a bit of luck, I guess, they can solve that problem and they can win a Nobel Prize. But a lot of Nobel Prizes are for discoveries. We set out to ask a different question, and we found something totally unexpected. And of course, you can't decide to find something unexpected. By definition, right? You can't decide to make a discovery. So that discovery was so spectacular and so intriguing that it took our whole life for the next 2 years or so. We became totally obsessed, and I've made other discoveries, as we do, and some of those discoveries have been moderately interesting to a number of people. Some of those discoveries may be interesting to about 5 people, but this discovery was a big discovery, and it really was absolutely challenging, totally fascinating, and a tremendous experience.
My name is Daniel Danko from Mora Friends Secondary School, Kiskunfélegyháza. And my question is, how has your Nobel Prize winning research influenced the researches and fightings against AIDS?
Well, our discovery really clarified how half of what we call the adaptive immune system works. There are 2 types of immune systems. There's the innate immune system, which is a kind of an early response system, and the Nobel Prize this year was for innate immunity. And 2 of the people who won it, one works with fruit fly, like Eric Wieschaus, and did knockouts in fruit fly. The other worked with mice and mammals and so forth. And so it shows you that innate immune system goes way back in evolution. Now, the adaptive immune system is really young. It's only about 400 million years old. It goes back to the jawed fish. And it divides broadly into what we call the humoral side, which is antibody, which is the basis of most vaccination. And we understood that for quite a while at a pretty good level. And the cell-mediated or T-cell system, and that was very poorly understood. Our discovery opened that whole field out in a totally different way and in a totally unexpected way, and it changed the way people thought about cell-mediated immunity. So that has, in turn, influenced anything that's been done since on T-cell-mediated immunity, and that's to do with transplantation, that's to do with tumor therapy, and of course to do with vaccine development in diseases like AIDS. And it's also analysed how we analyse these diseases, influenced how we analyse these diseases. So it was a very basic discovery that changed the whole field. It's as though a few of us, 2 of us, did a little experiment in a little lab off in Australia, and suddenly it becomes enormously expanded and so forth. So that's how you win a Nobel Prize. You make a little discovery somewhere that's kind of important, and then you get hundreds and thousands of other people to work on it, and then you get the Nobel Prize. That's in my book. That's in my book. Yeah, so it's good advice. I just can't tell you how to do it. That's
Would someone else like to ask a question? Yes, I see a hand there, please. Wait a minute, please. The microphone is coming.
My name is Ara Tahai, and I'm a medical student of the University of Szeged. I would like to ask you, how close do you think the medical science is in order to treat HIV or in order to produce the vaccine for this disease, which takes a lot of victims each year? How close do you think the medical science is to achieve this treatment or this vaccination?
Yeah, the story of HIV and AIDS is both a triumph for modern medicine and a lesson about our limitations. We've done wonderfully well with AIDS with drugs, with small molecules. So the biochemists, the pharmacologists who've designed molecules that target particular components of the virus replication cycle and so forth have allowed us to deal with AIDS. The problem with that is that people who have AIDS have to take 3 drugs a day in multiple doses, though that's getting a bit better, the so-called HAART therapy. And that's expensive. Not everyone gets it in the world, and it's a problem, particularly in the developing world, and so forth. And it's not great to take that many drugs a day, but if you do, you can live, or if someone does get AIDS, and the thing with AIDS is do everything possible to avoid getting it, of course, people can live a reasonably normal life. They're a bit more susceptible to some things, and they can have some odd conditions, but they can live reasonably normally if they get the drugs. Vaccine would be great because vaccines are our cheapest form of intervention. especially if we only have to give a vaccine once or every occasionally. But AIDS has pretty much defeated us as far as vaccines go. And the problem is that the virus changes all the time. So initially, if someone gets infected with the AIDS virus, the immune system handles it well. It controls it. There's a big fight goes on in the lymphoid tissue where the virus is growing between the T cells and the other cells that are growing the virus. But in time, the virus mutates away. It escapes. It keeps escaping. And as it escapes, it defeats the immune system and it progressively kills off part of the T cell system, the CD4 or helper system. When that happens, the whole immune system collapses. And when that happens, and that may happen after a year or 2 years or even 3 years, when that happens, then the individual becomes very immunosuppressed. And they usually die from something else. They die from tuberculosis or some of the organisms that sit on the surface of the gut or something. Now, many, many efforts have been made to develop AIDS vaccines. Hundreds of millions of dollars, billions of dollars have been spent trying to develop AIDS vaccines. And basically at this stage, we simply don't know how to do it. Now there's some strategies that are being tried, and for instance, strategies through structural biology, where people are trying to identify shared components on those viruses that can't change. Sometimes things can't change because they're necessary to form a particular structure or have a particular activity. They're trying to target the immune response towards that. But so far, there's nothing promising. So it's an enormous challenge out there, and maybe one of you guys will solve it. I don't see a solution at this stage, but it's enormously important we continue to work on it. The thing about AIDS is the HIV is not that infectious. It's only transmitted by blood or by sexual transmission. But if another virus came out of nature, let's say transmitted by respiratory infection, we'd all be in terrible trouble. So I think we need to know how to handle this virus, and we need to continue to try to develop a vaccine strategy for it. Though the drug side's great, but drugs are always a second best in that sort of situation.
Thank you very much. Before the next question of our students, I would like to ask you how you are coping with your jet lag, because you just arrived from Germany, but you were in India before.
And Australia. Well, the problem is we travel too much. We travel internationally. I think I'm permanently jet lagged. I seem to have recovered from jet lag, but I'm not sure if it's not that I'm permanently jet lagged.
All right. Do we have another question now to Mr. Doherty? Yes, I see the hand there. The microphone is coming to, and please, as quickly as you can do that. A little bit quicker, please. Very good, thanks.
I am Gergely Gajda from Bolyai Secondary School in Zenta in Serbia. And I would like to ask you if you have any inspirational message for us young students who want to learn science.
An inspirational message.
Yes.
Well, I think there are enormous possibilities in science. Everything opens out all the time. We ask one question, we do some analyses, we find new things. There are many, many new things happening. The application of molecular science, proteomics, all these new ways of looking very broadly at what's happening in biology are raising many, many, many new questions. To actually become a scientist, you have to learn your trade, you have to go to a good university if you can, work that teaches well, that has a good reputation, and then you have to, later, you have to do your research ticket, if you like. The research ticket is the PhD degree, the Doctor of Philosophy, and you try and do that with a good person. Then you will be starting to work your way into science. You may go away and do research elsewhere, or you may find a good mentor locally. You may come to it through different paths. You might come through it through studying science, through studying chemistry or physics or whatever it is that fascinates you, or you may come to it through studying something like medicine. And of course in Szeged we have many very fine medical scientists who first qualified as doctors. There are many paths to it, but the thing to ask yourself is, are you driven by curiosity? Do you want to find things out? Are you willing to find things out, like Dr. Huber said that would tell a Nobel Prize winner that they've got it completely wrong. In fact, that's one of the great things about science. Some guy of 22 or 23 can come along and they can tell some very famous person that, you know, okay, you just got it wrong. And it doesn't matter what you look like, it doesn't matter what sex you are, it doesn't matter what color your skin is. Science is a great level. It's an international culture, and it's something that allows someone with talent to actually emerge. And you can see that in the stories of great scientists. Some of them grew up on the plain in Indian villages. Some of them had privileged backgrounds. Others had very, very constrained backgrounds.
Thank you. Whereas in the fifth row, we've got, yes, please.
After I had read your autobiography, I saw your career is very unconventional because at secondary school you were interested in literature and history, but you knew you need a reasonable job, so you started your career as a veterinary student. So what do you think it is possible nowadays?
Oh, I think also things are possible. I've always been a writer, actually, but all my writing has been professional writing until fairly recently, until I've written a few non-fiction books. So writing has always been very important to me. I enjoy writing. I had a Yonath mention she'd like to write a novel. I actually have written a novel, but it's so absolutely dreadful that it's in the bottom drawer and it's never going to come out. And everybody who has read this novel, and that's only very few people, everyone hates it. Okay? So maybe sometime I write a better novel, but not this one. And so I'm forgetting the question. Basically, yeah, I trained to be a vet. I was going to go out I was going to do veterinary research. I had a cousin who was a researcher. He was studying insect-borne viruses in Australia. And he was a lot older than me, and I didn't really even talk to him about it, but I knew that example. So I knew there was such a thing as research and science. And so I decided I didn't want to be a doctor because I really didn't like sick people. I didn't want to spend time around people who are sick and whining and all the rest of it. And, you know, now I realize a lot of doctors don't like sick people either. And I could have been a pathologist, and then you only see sick people after they're dead. So I decided I was going to try to work on the diseases of food-producing animals. So I trained as a vet, but not as a vet who sort of looks after dogs and cats. I trained as the breed 'em, feed 'em, eat 'em school of veterinary science. And I was doing that. I did that for 9 years. I did research on diseases of cattle and pigs and chickens, and then I reckoned I needed to learn some new techniques. And so veterinary research is really very small when you compare it with medical research. There's not that much money, there's not that many people. So I went to a very well-known research medical school to learn some new technologies, which I was going to take back into that veterinary world. And that's when we made the big discovery that won the Nobel Prize, and I've been doing basic medical research ever since. I went from being a sheep doctor to being an MD, a mouse doctor.
Do we have another question? Or because of our time constraints, should I say thank you? Okay, then thank you very much for this excellent performance. And ladies and gentlemen, I'm sorry to have to say that Johnny Walker at the last minute was not able to join us, but we will introduce him. He has received the Nobel Prize in Chemistry in 1997, and the introduction to him is by Petra Jakus, Ságvári Secondary School, Szeged. Please welcome on stage, Petra. Thank you very much.
Ladies and gentlemen, it is my pleasure to introduce Johnny Walker. He was born in 1941 in Halifax. His parents were well educated and taught him many things about the world and the environment, so he became interested in nature at a very young age. At the age of 3, when he went to school, he was already able to read, and he was a very talented boy. He went to the local grammar school at the age of 11, where he learned mathematics, chemistry, and physics. He went on studying in Oxford at the university. He particularly liked organic chemistry, but soon became interested in genetics, molecular biology, biochemistry, and biology as well. In 1964, he received a BA degree and began to study peptide antibiotics with Edward Abraham, which he got his PhD for in 1969. He was also awarded the A.T. Clay Gold Medal in 1959 and the Johnson Foundation Prize by the University of Pennsylvania in 1994. He received many other prizes as well, but he got his biggest acknowledgement, the Nobel Prize, in 1997 for the exploration of the ATP formations mechanism. Several other research projects can be associated with his name that led to important discoveries. He analyzed the sequences of the proteins from the G4 and from the mitochondria. He also helped to uncover details about the modified genetic code in the mitochondria. He has published many writings about his experiments so that everyone can repeat his work, which is a very nice gesture. He married Christina Westcott in 1963, and they have 2 daughters, Esther and Miriam. He is currently the director of the Medical Research Council Mitochondrial Biology Unit in Cambridge. So please welcome John E. Walker, although he's not here.
Thank you very much. We cannot welcome him, but we can welcome Tim Hunt, who has received his Nobel Prize in Psychology or Medicine 2001. Please welcome him onto stage. And the introduction to him is by Renata Skita, representing Tancsics Secondary School, Orosháza.
Ladies and gentlemen, it is my great honor to take part in this occasion of the 75th anniversary of Albert Szent-Györgyi's Nobel Prize Award this afternoon, and it gives me enormous pleasure to welcome Tim Hunt. Tim Hunt was born on the 19th of February 1943 in Aston. In 2001, he was awarded the Nobel Prize in Medicine with Leland Hartwell and Sir Paul Nurse for the discoveries of key regulators of the cell cycle. His work led to the discovery of cyclins in humans and proved important in the field of tumor diagnostics. While he was working in 1982 at the Marine Biological Laboratory at Woods Hole, Massachusetts, using the sea urchin egg as his model organism, he discovered the cyclin molecule. He found that cyclins begin to be synthesized after the eggs are fertilized and increase in levels during interphase until they drop very quickly in the middle of mitosis in each cell division. He also found that cyclins are present in vertebrate cells where they also regulate the cell cycle. He showed that the cyclins bind and activate a family of protein kinase, now called the cyclin-dependent kinase, one of which had been identified as a crucial cell cycle regulator by Sir Paul Nurse. Tim Hunt joined Cancer Research UK in 1990, and the charity funded his research until he retired in 2011. And now, please welcome Tim Hunt.
As I realize you would like to add something, or not? As I realize you would like to add something to the introduction.
Well, it's not quite. This is about tumor diagnostics, or what is unfortunately not true at all. I've always felt a little bit guilty about being a cancer researcher and not doing anything towards curing cancer. I mean, I think I've learned a lot about cancer, but can't do anything about it. So. We did try, but it didn't work. It wasn't very useful.
But you can try it in the
future.
You have to
try. You have to try things, because you never know what's going to work. In my experience, anyway.
Okay, but let's hear the first question. Who would like to ask that one? Okay, please, here in the first row.
I'm Esther Tariani from Mora Ferenc Secondary Grammar School.
And I would like to ask that how do you spend your free time, if you have any, in order to become such a happy and smiley person?
Yeah. Well, the first thing to say is that people ask, how has the Nobel Prize changed your life? And the answer is, I think that everybody travels a great deal. And since I actually rather like traveling, I spend a lot of time in airplanes and stuff. But when I'm at home with my family, what I mostly like doing is cooking. And I've always really rather liked cooking, ever from a very early age. I'm not terribly good, but I really enjoy it. And I think the reason why I enjoy it so much is it's kind of like being a biochemist, and at a certain point you really stop doing the experiments yourself. But chopping up onions, I think, is a really good thing to do. Apart from that, I also, I sort of always, I was very pleased when computers came along, because I really I'm a great believer in gadgets, and I also love these little, ever since I was quite little, I used to take photographs. I had a very primitive box Brownie camera, and now you can just carry the thing in your pocket. I think it's absolutely great. So I think my iPhoto collection on my computer now has something like 40,000 photographs on it, so it's like a visual diary. But I really wish I could paint like Eric. I think that would be much, much better, because the difficulty with photography is that you can see, particularly with people's expressions, they're so fleeting. It's amazingly difficult to take a photograph of a person really animated and, actually capturing the essence of that person, because in a fraction of a second, their faces change. These moments are, whereas when you're a painter, you can sort of reconstruct them. Is that right? Kind of? Or change it, yeah, make it more real than it actually is in real life, yeah. So that's what I do. And now I'm retired. It's actually great, because I can do whatever I like. I go to my office most days when I'm at home, but I also go, I go to the Chinese supermarket, or I go to an art show, or whatever. It's wonderful not having to actually do anything. It's really great. Can't recommend it too highly.
Okay, thanks a lot for this answer, but let's move on to another question. Yes, please.
I'm Victoria Varga from József Attila Grammar School, Makó, and my question is, what are your aims for the future?
What are my aims for the future? Golly. Well, staying alive is the big aim. And no one's talked about this, but, I've had several near-death experiences. You probably haven't ever had have you ever had a near-death experience? No. Well, my first near-death experience was I was sitting in my room with a couple of friends, and I cooked them supper. And the supper consisted of lamb chops as well as other stuff. I don't remember what the other stuff was. And suddenly I found that I had a sort of lump in my throat, and then I discovered I was actually choking to death. But fortunately, I realized what was going on before I did actually die, and I sort of left the room and managed to blow out the lamb chop before I died. So that's why I'm here. So that was the first experience. But it shook me, actually, because I realized that I was actually quite close to death. And could have, if I'd breathed in hard, it would probably been the end of me. But I didn't. I mean, I realized, it was sort of a very curious sensation. A little bit worrying. Then the next time was I had actually been to a sort of a kind of student science fair where a friend of mine was giving a talk. And I'd taken my young daughter, Aggie, with me. And we were driving back, and suddenly as we were driving back, I was perfectly healthy, suddenly as I was driving back, I realized I felt absolutely terrible. I very rapidly came over with what felt like flu-like symptoms. And I went to bed and lay there all night, and all all the next day. And then we had to go, we have sort of two houses, a little flat in central London and a little cottage near where my lab was. And so we drove into town, and I was still, feeling absolutely terrible. And my wife, who often has sort of 8 o'clock meetings with doctors, mentioned my mystery illness to one of her, I think he was a hematology friend, and he said, oh, you should go back immediately and take him to the emergency room. And so I went back immediately to the emergency room, and it turned out that I was sort of suffering from septicemia, it was because when the bacteria were growing in my leg under my skin. I had a rather ugly rash there. So it was very funny. The Bulgarian doctor said, are you feeling ill? I said, yes, I feel like death warmed up. He said, well,
Maybe would you like to sit there?
No. And then he said, I'll give you some penicillin. So he got out some penicillin, and absolutely amazing. Half an hour later, I felt incredibly much better. It was quite extraordinary. But I don't know how close to death I was there. But it was a sort of lesson, And the third time, I'm a living medical marvel. The third time was not so long ago, about 3 years ago, I think. I had a very funny sort of bout of flu, or at least it felt like flu, but I didn't have any respiratory symptoms. And again, I went to bed. I was ill for a couple of days, and then I got up, and I remember going back to work on the 5th of January, feeling so happy that I was sort of well and going to, I was glad to get back to work and start experiments again and so on and so forth. And throughout that week, I sort of quite rapidly slowed down. The first time I realized it, that I was walking to a concert with a young friend, and I couldn't keep up with him. And I didn't really think very much about that, except that the next day I had to go to a sort of, it was a kind of a hearing about a planning permission from a neighbor. And I found that I could no longer walk up the stairs on the underground, which is something I normally always did. And by Friday, I could barely walk to the lavatory in the lab. And I met the storeman, Ronnie. And Ronnie said to me, oh, Tim, you do look rough, he said. You do look rough. He said, I think you should go to bed because you look exactly like what I looked like when I had my first heart attack, he said. So I took myself to bed, and my wife came back about 7 o'clock at night. And again, she rang up, and she was slightly worried because I was sort of And she rang up the National Health Service in England has a kind of dial-in. You can say what's wrong with you. And they said, go immediately to the nearest emergency room because this is pretty serious. I could barely walk. And the people said, oh, yeah, you've got atrial fibrillation and da-da-da-da. And so the next week I spent in the emergency cardiac care ward. And then a funny thing happened. So they didn't know who I was. And so they sort of, a gang of girl doctors came in and said, would you like some aspirin? I said, why would I want to take aspirin? It's for the pain. I'm not suffering from any pain. It's just that I can't breathe properly. Anyway, eventually, then they discovered, somewhere about Thursday, they discovered who I was, and suddenly the standard of care improved dramatically. So maybe it is worth winning a Nobel Prize. Anyway, so now I have to, and then it turned out I was a very standard case, but there was sort of, damage to my heart, and it still doesn't beat properly, so I still can't walk as fast as I would like to be, and I have to take lots of pills every day. So. But it's a sort of funny business. You realize that you're mortal, and there my phone is ringing. Who is this? No idea. Somebody from Oxford. Maybe all right. Hello? Yes. Listen, I'm at a student question and answer in Szeged. So I think I'll call you back. Oh, you're going to do it? Yes. Something like that. Oh, I'll see you there. Fantastic. On the same plane as I am, so that's very good.
Excuse me for asking
Sorry,
that. Maybe I don't hurt you, but a lot of people think and sometimes say that to become such a successful scientist as you are, a person has to be a little bit crazy. Do you agree with that?
No, I don't think so. Look at this fine company of men here. I may be crazy, but they're not.
Okay, it was only just a joke. Let's move on to another question. Who would like to ask?
Anybody else?
In the fourth or fifth row, I see.
Hi there.
Hi.
My name is Vladimir, and I'm trying to study medicine here in Szeged. So my question is, what do you think are the greatest scientific achievements in the 20th century?
The greatest scientific...
But you can name a few.
The greatest scientific achievements of the 20th century. Boy, oh boy. Have you ever been asked that before? I've never been asked that before. Well, let's have a stab at it. What are they? Well, there's the discovery of X-rays. That was pretty important. Wouldn't you say? I think so, yeah. Because, I mean, and interestingly, you see, that's a very good example of a discovery. Nobody knew that X-rays existed before they were discovered. Right? And the discovery of X-rays and the discovery of radioactivity absolutely opened the windows on a wholly unsuspected world, which, of course, I mean, it happens to have medical achievements, but I mean, much more important, it has, you know, we have light and power and all that stuff. I personally think that the revolution in physics that happened mostly between the wars, I mean, the development of quantum mechanics is a quite extraordinary achievement of the human mind, largely because I can't understand it a bit. I mean, I suspect that Robert does understand it a bit, but I certainly don't. So that's one thing. I think the other thing is that in my lifetime, I think that what biologists have come to understand is quite extraordinary, and I never believed when I started out being a biologist, which I always, by the way, wanted to do, that we would know and understand so much by the time that you know, my career was effectively open. I mean, I think I particularly admire what Eric and Yanni did, because I think that's an absolutely monumental achievement. I mean, everybody wanted to understand how development worked. Nobody had a clue, really, and now we have a... Well, I mean, we still... If you hear Eric's talk tomorrow, he'll tell you we still don't have a clue, but actually we do. So, and then, you know, all this, the invention of penicillin and the antibiotics, I think, you know, I mean, the conquest of disease, and I suppose really more importantly, the conquest of hunger. I mean, you know, none of us ever go hungry these days except by our own conscious will or disease, you know. I mean, it's just absolutely amazing that, whereas I think in years, you know, only sort of 500 years ago, I bet you everybody was pretty hungry most of the time, actually. So, I mean, you know, we're very lucky to live, I think, in this age, and it's largely because of the fruits of science and technology, I suppose.
Okay, thank you. I think we are running out of time, so
yeah,
I would like to suggest
to
maybe a quick question to you, if somebody would like to ask. Yes, in the first row, okay? Okay, and the last question, but a quick question, please, and a quick answer, okay? Thank you. Rest.
My name is Kinga Boros from Móra Ferenc Secondary Grammar School, and my question, what would you be if not a scientist?
What would I be if not a scientist? I don't know, because I always wanted to be a scientist, actually. So, you know, I don't. I've always been a scientist. So I never contemplated any alternative career, actually. Good.
Okay, the last question, because I promised that you can have the last question.
Okay.
Where are you? Sorry.
I'm over here.
Oh, right at the back. I see.
Yeah. As we all know, a lot of the problems in the world right now involves like economy and debt, and unfortunately for research, you need money. Do you think this has or will affect the chances of getting grants, or is it just based on each country's policy?
Well, yeah, no, I know a little bit about this. I mean, you know, the truth of the matter is that scientific research is a luxury. And it's a luxury that many poor countries can't and arguably shouldn't afford. But I don't want to get into that because economics is not a science, it turns out. I mean, the economists just haven't got a clue, as you can see. Right? They can't control anything. So I think this is a question that we really ought to sort of discuss in private afterwards.
Okay.
But you know, I mean, the problem, I think, basically is that because of the nature of discovery, which has been well explained by my colleagues, it's a rather random process, and it's totally, by its very definition, unpredictable. This makes it awkward for politicians and tax gatherers because you can't... I can pay for a voyage of discovery, but I can't guarantee that you will find the new world at the end. That's the problem. So you need an awful lot of spare cash. It's a chancy business. It's a lottery, basically, scientific research, I would say.
Thank you very much, and thank you very much for your answers. Ladies and gentlemen, Tim Hunt. Okay, and ladies and gentlemen, we would like to introduce Ciechanover, who has received his Nobel Prize in Chemistry in 2004. Please welcome him onto the stage. And the introduction to him is by Krisztina Földi, Bibó Secondary School, Kiskunhalas.
Good afternoon, ladies and gentlemen. First of all, allow me to jump at the chance and admit how glad I am to have the opportunity to introduce you a great biochemist, Professor Ciechanover, who, with Avram Hershko and Erwin Rose, was awarded a Nobel Prize in 2004 for the discovery of the ubiquitin-mediated protein degradation. Professor Ciechanover was born in Haifa in a post-Soviet Israel in 1947. He was particularly interested in biology and carried out experiments from a very young age, at the age of 11. He studied the elementary and high school in his hometown in Haifa, and later went on to attend Hadassah and the Hebrew University School of Medicine in Jerusalem. He participated in postdoctoral training in the USA, in Massachusetts as well. Though first he wanted to become a practicing physician, after examining patients he doubted that this career would satisfy him, and so he decided to become involved in the basic research in the field of biochemistry instead. In 1972, Professor Ciechanover met Avram Hershko, who was born in our country in Hungary, in Kartag. And 4 years later, they collaborated with Erwin Rose as well, started their research that led to the discovery of the ubiquitin system. By studying the mechanisms of energy-dependent protein degradation, they found a system that used a type of DAS label together with 3 different enzymes to attach it to the proteins to be destroyed. We know now that controlled protein degradation regulates other very important processes in the cell as well. Examples are the cell cycle, the repair of DNA damage, and immune defense. Among other things, this knowledge can be used to produce new medicines against various diseases. Nowadays, this Nobel laureate is a research professor in the Department of Biochemistry at the Technion Rappaport Faculty in his hometown in Haifa. Please welcome Professor Ciechanover with great applause.
Thank you very much for the introduction. And then I would suggest to move on to questions now. Who would like to have the first question? Yes, please, in the second row. We can give a microphone to you.
Well, my name is Anna Lorenz, and I would like to ask, what would you advise for youngsters who want to be researchers? Advice for youngsters who want to be researchers.
What do I want? What advice?
Advice.
Advice. Wow. This was asked several times before. I would repeat, actually, it's the very end, so you heard almost everything. And I think that the best thing to do is to go out of your feeling and to do what you really like to do. And as Ada said, if you will convert your profession or what you are doing daily into your hobby, or the opposite, then you'll be very happy. I think that for myself, for my own career, I think that I have undergone this decision-making because I was not only trained as a physician, but I then started my practice as a surgeon. And I spent several years in the operating theater. And then I decided that somehow I didn't feel that it will carry me for the next 4 or 5 decades, whatever I was left to live at that time. And I decided to start all over again. And Avram at that time returned from a postdoctoral fellowship. I was one of his very first students. And I decided that I want to do what I want to do and what I feel good about. And therefore, here I am today. And if I had to change in the middle again, because I wouldn't have liked it, I would have changed it again and again. And I think that it belongs to a more general philosophy that life is in many ways purposeless. You don't have a purpose in life and then you achieve it and then you are resting on the wreath. I mean, life is a collection of interesting experiences and you are learning forever and you are collecting more and more experiences because anyway, in any lifetime we shall not achieve, anything. We are not coming to an end. We shall not achieve infinite knowledge of any problem, solve a problem to its very end. We are just thin layers. We are adding a layer of knowledge and then some other people will pick up on it and continue and continue and continue. So therefore, you should do what you are really happy with. Let it be science, literature, music, sculpting, architecture, whatever you feel good in.
Okay, thanks a lot. Yeah, let's move on to the next question.
I'm Máté Iványi from Móra Ferenc Secondary School, and my question is, which was feeling when you recognized your discovery or when you received the Nobel Prize?
Well, obviously the discovery. The question was, when did I feel better, when I did the discovery or when I got the Nobel Prize? First of all, when you make a discovery, it's not a one-day discovery. It's not a eureka. It takes years to realize what is the discovery because initially you suspect that it's wrong and then you say, okay, there is some phenomenon, but there must be a mechanism. So let's first find the mechanism and the enzymes and crack down the molecular details. And then, even then, you don't understand how important it is. It's a life story and it's still, unraveled daily. I'm still in the lab. I have a wonderful group of people working with me. Now, jokingly and humoristically, obviously we are not going to appeal the decision of the Nobel Committee or to reject it, but at the end of the day, the purpose in life is not to get prizes, and with all honesty. And you get the prize and you are happy, and the next day you are happy, and then the next day you are happy, but at the third or fourth day, you have to go back to work. And that's what you are doing. And there is so much you can celebrate, and therefore, it's an event. It's a happy event of recognition. People, let's admit it, people need recognition. It's part of human nature. But that's not really the purpose of life. I think that discovering, cracking down the secrets of nature and seeing it even becoming useful for human beings. We didn't predict it. I'm not studying, I'm not developing drug by myself. But then seeing how other people pick it up and develop drugs, and then meeting patients and being associated with patients, you are seeing a whole world unraveled and opened up in front of your eyes. I mean, how can anything rival it? I mean, this is such a fantastic feeling. And prizes are events in life. They are scattered, time-defined events. They are not filling your life. So obviously the answer, and with all sincerity, is that being involved in science and in discovery and cracking the secrets of nature is an unbelievable source of happiness and joyfulness.
Thank you. Who would like to ask the next question? Yes.
It's late and you're all hungry and you don't have to feel obliged to ask any questions.
My name is Melinda and I came from Orosháza, Táncsics Mihály Secondary School. And my question is that in your childhood, could you ever imagine that you would receive the Nobel Prize?
No, again, it belongs to, in my childhood, I couldn't even imagine what I'm going to be. And I'm still not sure what I want to be when I shall grow up. You see, I started with medicine, and I grew up in a small country in Israel. You're not sure about your future. These days we are dealing with the Iranian A-bomb, so you're uncertain. There is a lot of uncertainty. My parents came as refugees from Poland, being chased by anti-Semitism, and you're always growing up on some kind of, unsafe soil, and you don't know. And then, like anybody else, I served in the military. I was an officer in the Israeli military for several years, fought myself several wars, mostly the 67. The most remarkable are the 67 war and the 73. So many of my friends being killed in the battlefields, and so a lot of blood. And you really, it's a little bit hard to plan, and therefore I told you, but it's true also for people who are living on more safe grounds. Life is purposeless. I mean, I don't believe that there is an aim in life. I'm just following my feeling and do what I really like to do, and I like to serve my country, and I'm part of a country, I'm part of a nation, and I'm very proud of it. It's a young nation and very successful one, scientifically, technologically, and therefore I didn't really plan, and I decided I was always after biology, and I thought that medicine will satisfy me, and I started with medicine, and then I was a military physician, but then I started surgery, and then I decided it's not for me, and then Avram returned. You know, it's a lot of coincidence around it, and then things you just let things evolve. So I was involved in another career, and like you ask Tim what he would have done if he wouldn't have been a scientist. I had a profession already, and I thought I was successful, and I even liked it, but then I decided to do something else. So there was not a decision that was taken at an early age, the choice of my career. I mean, I just let it go. I just let it go. I'm not a long-term planner in that sense.
Actually, what do you think? What are the most important skills to be such a successful scientist?
Well, I must admit and adhere to the truth of history that the idea of working on protein degradation was not mine. It was my mentor, Avram Hershko. He had this idea, and he himself took it from Gordon Tompkins, a mesmerizing figure in that field that was interested in how enzymes of gluconeogenesis are being degraded following removal of the induction. So people ask me, how did you come to the idea? I said, I was free from it. I mean, it's not mine. But I think that the most important thing for success in science is really passion. I mean, an endless amount of passion. You know, science really makes me shivering. I mean, the laboratory, looking at experiments, looking at data, discussing data with the students nowadays, at that time with the mentor, was really an unbelievable experience, and I loved it more than anything else. I don't tell it to my wife, but I really loved it more than anything else. It's an unbelievable amount of passion. I was living in the lab. The lab was my home, and it's now even my home. You know, I think that the beauty of being in science and loving it, that it's not a job. Again, it's a hobby. You're not going to work in the morning and come back home in the afternoon. You're going to do something that you jump out of your bed in the morning, and that's what you want to do. And you go to sleep with it. And for me, 40 years into career or 30 years into my career, every day is a new day. I mean, every day in the lab is a new day. And I really love it. I'm never bored with it. And it really drives me. And I think that this is really a source of success. Then there is a transition. And that's becoming independent. Because initially, you have a role model. You are studying with somebody, like an apprenticeship. You are seeing Avram is a type of a teacher that he never talks. I mean, you can never, he will never tell you anything. I mean, he's silent. But you can follow him. It's like in surgery. That's what I learned. In surgery also, good surgeons will never tell you what to do. You follow their hand. And if you have the talent in the hand, holding the scalpel, never lift it up, making the whole cut in one shot, not damaging the tissue, you got it. It's apprenticeship. You just look and follow. And I think that an important thing in science is becoming independent. I had my own idea. I mean, I didn't discuss it with anybody. I carried it out. And for that, I had a great opportunity when I was a postdoctoral fellow at MIT with Harvey Lodish, and Harvey let me alone. I just was left alone. In the morning, I did what he wanted me to do, but as of noontime, all the way to midnight, I did what I wanted to do. And that's working on the ubiquitin system, partially alone, partially in collaboration. I think that this is the moment of elation, that you had an idea and it's right. And nature just tell you, you were right, you guessed right what's going on. So I think that there are stages, and that adds to your passion, that now you're on your own and can do it, can drive the car on your own without any guidance. So there is development, a slow development in being a scientist. And then, you're collecting a group of talented people around you and you work with them. And it's huge fun. It's a huge fun. It's hard to describe in words, but it's fantastic to do it.
Thank you very much. Could we have another question? Maybe? Yes, thank you.
Hi, my name is Barnabas Tóth, and I would like to ask whether, haven't you ever wondered to write a book about this topic, about the relations of science, because it sounds great?
To write a book?
Yes, about this topic.
Maybe at the age of 95, if I'll get there.
If you can manage.
I'm too busy to write a book. Writing books for me is like looking at the past, and maybe it's important. I'm not an out. Let's take it seriously. It may be important to write a biography with experience, because my biography has some points in it. Again, coming from a small country, not too much resources, parents that escaped Europe, and so on and so forth. So I may think of it, but for now, I'm feeling that I'm being chased by science. I feel another 15, 20 years, I don't know, of hopefully healthy life. As Tim said, the most important thing is to remain alive and healthy, not only alive. I know some people that are alive in the intensive care units. They're not so happy about it, if they can tell. But I feel that science is developing so fast. What's going up around us is developing so fast. New methodologies of imaging, sensitivity, high sensitivity proteomics came to the world. Now we can look at the cells from above and seeing dynamically what's going on, that I feel that more now I have even less time than I had before. Not only because my lifetime is shortened, but also because science expands exponentially. And I really want to catch up on it. I mean, I feel that I want to drive this car as long as I want. And writing books, maybe I come one day to the point that I feel that I'm not good enough anymore to educate the young generation, which may well happen. That you take a huge responsibility with the young generation, because when I have graduate students and postdoctoral fellows, I'm responsible for their career. I mean, I'm fully responsible for these graduates, to settle them up, to set them down in a good postdoctoral position, and for the postdoctoral fellows, I'm responsible for their position and for their future career. There may come a point that I feel that, you know, I don't want to take this responsibility anymore, I'm outdated, and then I'll find time to write a book with some points of my biography, which I find interesting, but now I think that science prevails.
Thanks a lot. We've got time maybe for one more question. Who would like to have the last one? Yes, in the last row, please. In the middle of the last row.
What do you think is the most important thing that scientists should have? Keep in mind. The most important thing that scientists keep in mind. What is the most important thing that scientists keep in mind, should keep in mind?
The most important thing that scientists keep in mind, I think that I stressed it and others stressed it as well, is really passion. I mean, if you don't love it, don't do it. But I think that it's true for every profession. I mean, you must love what you do. And you have to close your eyes and to ask yourself, am I really happy in what I'm doing? And really report to yourself in most honesty and truthfulness whether you are really happy in what you are doing. And the rest is not important. I mean, the rest is really not important. That's what I feel. And science is truly a privilege. I'm a little bit disagreeing with a team about small countries doing science, because taking example from my own country, I mean, fostering and nurturing the university and the higher education system and the science and technology made Israel what it is, because at the end it's a great bank, you know, investing in science, and you know, every dollar you put in, you get a lot back. But you're right, there is probably a lower threshold from which countries has other priorities, and they cannot afford this. But it's not. It's truly not a luxury, because for Israel it's a matter of existence. I mean, without it, we wouldn't have existed, surrounded by whom we are surrounded. So I think that again, it's passion, and also, you know, everybody grows up in a different environment. Hungary is not Israel. Israel is not Germany. Germany is not the UK, and so on and so forth. But for me, it was also being an Israeli. I'm very proud of it, and I feel that I contribute to my country. So, you know, besides the central avenue that was mentioned by Peter Doherty, science is an international culture, and it's truly an international culture. You see here a collection of people coming from different cultures, religion, faith, countries, did different things in their life, and they immediately can find common language and talk immediately to one another. Nevertheless, there are lines of separation that take us apart, and I think that they are important as well. And altogether, they build what we are. And I think that Hungary has a huge tradition in science, going back to the many busts that you see here in the neighboring room, from Edward Teller to Szent-Györgyi, that we are celebrating the 75th anniversary of his Nobel Prize, to many others, von Neumann, other people that didn't get the Nobel Prize, and Wigner, and many others, and they bring a lot of pride to this country, being originated from this country. So a country also takes pride of its own sons and people. So this is also something which I see very important. So since this was the last question, let me just wish you all, because the young faces are really fantastic, a great success in whatever career you're going to choose. So have a wonderful weekend. Thank you very much.
Thank you very much. Thank you very much again. Ladies and gentlemen, we expected that Professor Andrew Schally will join us too, but unfortunately was able to do that. But we will introduce him too, as normally. The introduction to him is by Otto Welles, who represents the Bolyai Secondary School from Zemca. Please welcome him on stage.
So, well, actually, to begin with, I think I owe you an apology, because, after seeing the scientists here who handle the situation with such ease, I don't think that I can, be serious anymore, but, I will try my best. So, just for a record, as, of course, everyone here, I'm quite delighted, and, of course, honored to be here, and to be given the opportunity to introduce Andrew Victor Schally. He is a well-known scientist in endocrinology. So actually to be here and talk about him is like a privilege like no other. So to begin with, Schally, or Andrew, if you may prefer, he was born in Wilno, in Poland, on, I think, November the 13th in 1926, as a child of a professional soldier and a noble woman. So when the Second World War broke out, Schally fled from Poland to Romania, and thus he managed actually to survive the Holocaust. At the age of 19, right after the end of the World War, he moved to England and Scotland to receive an education. At the age of 23, he joined the National Institute of Medical Research in Mill Hill, that's in London. And in 1952, he moved to Montreal, Canada, and there he was given the opportunity to learn endocrinology at McGill University. And 5 years later, when he received his doctorate, he stepped on a path filled with researches. And in 1961, he visited the Institute of Biochemistry in Uppsala. And later, he went on to gather experience, about electrophoresis and the gastrointestinal hormones. And a year later, he became a neutral citizen of the United States, and he also became chief of the endocrine and polypeptide laboratories. So, along with Dr. Blesser, Bowers, Barch, and Locke, he established a laboratory in New Orleans, and there he, along with his colleagues, solved a wide variety of problems revolving around the gonadotropin-releasing hormone. And actually, the following years, after researching the gonadotropin-releasing hormone, they focused on the LH and RH. And in 1971, actually, they managed to solve this problem. And right after that, they focused on the PIF and GHRF. And actually, in 1973, they managed to purify this hormone. And also, they completed structural researches and managed to synthesize it. Actually, in the same year, he received the rare title of Senior Medical Investigator. And as we all know, in 1977, he won the Nobel Prize for his studies about the brain peptide hormone production. And that's all.
Thank you very much. Ladies and gentlemen, I would like to thank all of you for your attention and participation. Thank you very much for your wonderful questions. And of course, thank you very much for your wonderful answers. I think it was a really very enjoyable discussion. And may I ask all of our Nobel laureates and the students who made the introduction to come up onto the stage to have a picture commemorating this really special event. If I can do that, I would like to ask you to come up onto the stage. And all of the students, please come up to the stage too to have a picture. Thanks a lot for your cooperation. And if you don't mind, I would like to ask the 27 previously chosen students who attended at the contest to come up onto the stage, because it's a very special and rare moment to have a picture with our Nobel laureates. But please, very quickly, because our Nobel laureates maybe will lose their patience. historical moment. You never know, maybe among our students are some Nobel Prize winners in the future. We will see. Okay, thanks a lot one more again for your participation, and thanks a lot for your attention. Have a nice day, and I really hope that you enjoyed this open forum discussion. Have a nice day. Bye-bye.
The film was commissioned by the University of Szeged. The project is supported by the European Union and co-financed by the European Social Fund.